Both B cells and T cells carry receptor molecules that recognize specific targets. T cells recognize a non-self target, such as a pathogen, only after antigens, small fragments of the pathogen, have been processed and presented in combination with a self-receptor called the major histocompatibility complex, MCH, molecule. There are two major subtypes of T cells, the killer T cell and the helper T cell. In addition, there are suppressor T cells, which have a role in modulating immune response. T cells only recognize antigens coupled to class I MCH molecules, while helper T cells only recognize antigens coupled to class II MCH molecules. These two mechanisms of antigen presentation reflect the different roles of the two types of T cell. A third, minor subtype, are the Y omega T cells that recognize intact antigens and are not bound to MCH receptors. In contrast, the B cell antigen-specific receptor is an antibody molecule on the B cell surface and recognizes whole pathogens without any need for antigen processing. Each lineage of B cell represents a different antibody, so the complete set of B cell antigen receptors represent all the antibodies that the body can manufacture. Killer T cells. Killer T cells are a subgroup of T cells that kill cells that are infected with viruses and other pathogens, or are otherwise damaged or dysfunctional. As with B cells, each type of T cell recognizes a different antigen. Killer T cells are activated when their T cell receptor, TCR, binds to this specific antigen in a complex with the MCH class I receptor of another cell. Recognition of this MHC antigen complex is aided by a co-receptor on the T cell, called CD8. The T cell then travels throughout the body in search of cells where the MHC1 receptor bear this antigen. When an activated T cell contacts such cells, it releases cytotoxins, such as preferin, which form pores in the target cell plasma membrane, allowing ions, water, and toxins to enter. The entry of another toxin called granulysin, a protease, induces the target cell to undergo apoptosis. T cell killing of host cells is particularly important in preventing the replication of viruses. T cell activation is tightly controlled and generally requires a very strong MHC antigen activation signal or additional activation signals provided by helper T cells. Helper T cells. Helper T cells regulate both the innate and adaptive immune responses and help determine which immune responses the body makes to a particular pathogen. These cells have no cytotoxic activity and do not kill infected cells or clear pathogens directly. They instead control the immune responses by directing other cells to perform these tasks. Helper T cells express T cell receptors, TCR, that recognize antigen bound to class II MHC molecules. The MHC antigen complex is also recognized by the helper's cell's CD4 co-receptor, which recruits molecules inside the T cell, e.g. LCK, that are responsible for the T cell's activation. Helper T cells have a weaker association with the MHC antigen complex than observed for killer T cells, meaning many receptors, around 200 to 300, on helper T cells must be bound by an MHC antigen in order to activate the helper cell, while killer T cells can be activated by engagement of a single MHC antigen molecule. Helper T cell activation also requires longer duration of engagement with an antigen-presenting cell. The activation of a resting helper T cell causes it to release cytokines that influence the activity of many cell types. Cytokine signals produced by helper T cells enhance the microbial function of macrophages and the activity of killer T cells. In addition, helper T cell activation causes an upregulation of molecules expressed on the T cell surface, such as CD40 ligand, also known as CD154, which provide extra stimulatory signals typically required to activate antibody-producing B cells. B cells. A B cell identifies pathogens when antibodies on its surface bind to a specific foreign antigen. This antigen-antibody complex is taken up by the B cell and processed by proteolysis into peptides. The B cell then displays these antigenic peptides on its surface MHC class II molecules. This combination of MHC and antigen attract a matching helper T cell, which releases lymphokines and activates a B cell. As the activated B cell then begins to divide, its offspring, plasma cells, secrete millions of copies of the antibody that recognizes this antigen. These antibodies circulate in blood plasma and lymph, bind to pathogens expressing the antigen, and mark them for destruction by complement activation, or for uptake and destruction by phagocytes. Antibodies can also neutralize challenges directly by binding to bacterial toxins or by interfering with the receptors that viruses and bacteria use to infect cells. Altogether, these cells work hard to prevent infection from recognized foreign invaders.